Hello, I'm Phil Rogowski, the director of the Maryland Film Festival. Welcome to our BioTech event. This is the third year we've done a event specifically focusing on biotech. And biotech covers a lot of areas. Whenever I invite organizations to join this event, they ask me, please define it, because as I just mentioned, it covers a lot, and it is an ever-changing and ever-growing field. And we are very fortunate to have three companies and three companies, organizations to talk to us about different areas of biotech. So I'm going to turn over our program to someone who knows much more than I do, to Travis, who will be talking about proteins. Travis, please take it away.
Can you hear me?
We can hear you well, yes.
Okay, great. Yes, welcome. I'm here in my lab, and most of what I do is related to proteins. And since nowadays everybody is focused on fighting the coronavirus, I'm very interested in the coronavirus proteins and also in human antibodies, which were already a big thing, and now they're a much bigger thing. So let's talk about this virus. I have a model that I'm going to use here is a Lyme. I hope you can see this. So the coronavirus is basically each coronavirus particle is sort of round, and it has a bunch of these proteins sticking off of it, which I'm going to represent by these screws. So I hope folks can see that. On an actual coronavirus particle, there would be about 70 or 80 of these spikes. And that spike is the protein that gets the virus inside of human cells. So it's a very wicked and very devilish protein. It's made with a part on the outside that is ready to stick onto human cells. It's got a surface that is just right to bind onto a protein of our cells. And then the other thing about this coronavirus spike protein is that it's evolved to work like a spring, so that after it binds a human cell, it snaps, and that pulls the human cell, which on the scale of this Lyme, if this was a virus, a human cell would be about the size of this box. So the human cell is much larger. But once it sticks, then the snap pulls the virus, it smashes it into the human cell, and it fuses and dumps its contents into the human cell where it can get to work making more viruses. So there's about 20 different proteins inside here, but the spike is the one that sticks out, and that's the one that the vaccines are going to be working against. So there's a lot of work nowadays to take the spike protein, and you can take the spike protein all by itself, or you can take a kind of mashup of the virus and use that to make a vaccine against the coronavirus. So that's one strategy that I'm sure you've heard about. Now, here in my lab, most of what I do is about protein structure. And the other protein that's very important in this work is the antibody. So this is a model of an antibody that we made on a 3D printer. So it's a famous shape with like a Y-shaped molecule. And that's because these two top ends bind onto something, and every antibody has its own specific target. So in our blood, we have millions or billions of these floating around, and a few of them might be able to bind onto the coronavirus. And the part that they're going to bind is the spike. So there's a lot of work nowadays in biotech to try to find antibodies that have just the right sticky part here to stick onto the spike protein. And then the antibody has other capabilities that will cause the immune system to ramp up and fight away this virus. So we're looking for good antibodies, and that's what Regeneron is. Regeneron is a biotech product right now that's a candidate medicine, and there's a lot of other antibodies that might be the medicines of the future against coronaviruses. So that's kind of my angle on this in a nutshell. And just in terms of biotech, I just wanted to share that a lot of what biotech people do these days is they use tools like this, the pipette, and they use some kind of plates like this to do lots of tests. You might have 24 or 96 wells, and you might be testing 96 antibodies, or maybe you're helping to test people for whether they have been infected or not. So then you might be able to test 96 people at a time, 96 samples at a time. So there's a lot of interest in biotech in making things efficient, making them small, making them fast. And a lot of that is going towards using robots, so that I'm going to show you a robot here in my lab. I'll just turn my screen around and see if you can see this instrument there that I'm pointing at. It's just you can see it's kind of like it's got a lot of tubes and bottles and very small tubes, and it's able to be programmed so that it can do a lot of science overnight without a human there. It can produce a lot of information, and that's one thing that they're developing now to do high throughput testing for the virus, and also high throughput evaluation of antibodies to see which ones are going to be effective as medicines. So maybe that's well, let me just tell you one more thing, is that the work that I do is about protein structure, and we're trying to find out how proteins work, how they can be engineered, and the way that there's different ways to do that. And one way that I use is to grow crystals of proteins. So a crystal might have a shape like this, kind of like a gemstone, but it's actually made out of trillions of proteins, maybe about the size of a grain of sand, the crystal. But if we can grow a good crystal, then we can use that to find out the structure of the protein, and then how the protein works, and how we might be able to make it better or understand the biology better.
Travis,
that's it.
Travis, would you like to explain where you work?
Oh, I work at the Institute for Bioscience and Biotech Research, which is here on the campus of the University of Maryland at Shady Grove. And this is a kind of a collaboration between the university and the National Institute of Standards and Technology, because we're all interested in the science and also in the basic measurement skills that are required for science.
And what is your background? How long, what is your training? What level of?
I got a PhD in chemistry. I was always interested in the periodic table. So, and how these 100 elements make up the whole universe, apparently. So I studied chemistry, got a PhD in chemistry, and then I did a postdoctoral program, and then I started working here.
And how long have you been working there? How long have you been working there?
I guess about 30 years.
Okay. All right. Well, thank you very much. That was very interesting. And if anybody has any questions for Travis, please put it in the chat box and we will relay it to him so he can get it answered for you. So thank you so much. We're going to move on to our next presenter. All right. Sam, are you ready?
Yes, let me know if you can see my screen.
We can see your screen, Sam.
Okay. Thanks, Phil. Thanks for the opportunity to present. And Travis, thanks for sharing some pretty fascinating work you guys are doing there. So I'm going to talk to you guys just a little bit about my organization and the work that we do. Work for Lung Bioengineering Incorporated. We are wholly owned subsidiary of United Therapeutics Corporation, which is headquartered here in Silver Spring, Maryland. And the company is really dedicated to finding a cure for pulmonary arterial hypertension. In addition to that, we're heavily invested in the work with organ transplantation. Our ultimate goal is to be able to find a way to provide an unlimited supply of organs for transplant for patients that are waiting on the transplant list. So there's a huge unmet need for patients that are waiting. So specifically what I'm going to talk to you about is our focus on lung transplantation and the work that we're doing to help patients that are on the transplant list. Before we go into there, I'd like to share just a few statistics when it comes to lung disease in the United States. So chronic lung disease affects about 35 million Americans. And unfortunately, that results in a little over 160,000 deaths every year. When we look at patients that have chronic lung disease to get to the point where their lungs no longer function, they potentially could be listed for a lung transplant. And unfortunately, when you look at the numbers that we have for lung transplantation, they're not very high. In 2019, there were only a little over 2,700 lung transplants. And out of all the organ donor patients in the year 2019, only about 23% of the lungs were used for transplant. So it's a pretty glaring statistic. There's 3,000 new patients that are added to the transplant waiting list every year. Some of the reasons why patients may be listed for a lung transplant include chronic obstructive pulmonary disease, which is COPD. And if you guys are familiar with that, you know, the way to get COPD is by smoking. That's what leads patients to eventually get to the point where they have COPD. IPF is idiopathic pulmonary fibrosis, and that's where the lungs become scarred, but we really don't know the reason why. And as a result, they lose their elasticity, and patients have an issue exchanging gas and keeping their blood oxygenated. There's also cystic fibrosis, and then the disease that's more near and dear to our organization's heart is pulmonary arterial hypertension, which, you know, we have drugs that our company manufactures for PAH, but if for some reason those drugs stop working, the patient may have to be listed for a lung transplant. So the work that we do, we have two facilities, one in Silver Spring, Maryland, another facility in Jacksonville, Florida. And the work that we do is related to lung transplantation in that we work with lung transplant surgeons across the US who evaluate lungs for potential transplant and direct lungs to our facility, either in Silver Spring or Jacksonville, for additional evaluation. So if they're potentially considering transplanting one of their patients with a set of lungs and there's some questionable function, or they think that there's a possibility that the lung may not do well in the recipient, what they'll do is they'll take those lungs out, they'll send it to our facilities, and then our team works on those lungs for a period of 4 to 6 hours, where we get additional information for the transplant surgeons. And if they meet certain benchmarks, then the lungs are packaged up and they go to the transplant center where they're eventually transplanted into a recipient. So when they come to our facility, we work on them outside the body. So it's kind of unusual. We put them in this plastic dome, sterile dome that's in the center of the room, and we connect the lungs to a circuit system that keeps the lungs flowing with a bloodless solution. We control the temperature of the lungs, and we also will hook them up to what's called a ventilator, which allow these lungs to essentially breathe inside of that dome. And during the time that they're inside of that dome, we're constantly evaluating the function of the lungs. We're collecting samples from this perfusion system to see how well the lungs are oxygenating. We're maintaining the temperature. And during this time, all the data that we collect is transmitted to the transplant center, who is looking to make a decision on whether the lungs will go for transplant. So the way the process works is at the donor hospital anywhere in the United States, you may have an organ donor patient that's being evaluated where the lungs may be questionable. The lungs are then removed from that donor at that donor hospital. They're sent to our facility here in Silver Spring, Maryland, or in Florida. We work on them for a period of 4 to 6 hours. And then if the decision is made to move to transplant, we package them up and then they go to the recipient hospital. So from the time the lungs are removed from the body to the time we have to have them on the system, is limited to 10 hours. And then from the time that we stop the procedure in our facility to the time they have to be transplanted to recipient is 6 hours. So it's not a lot of time. So the way the lungs travel throughout the US to these centers with recipients that are waiting is we charter private jets that take them to wherever they need to go. They fly what's called lifeguard. So they take off and land before any other plane. And the only one that trumps them is Air Force One. A little bit about what we do. So I'm going to show you a little bit some of the pictures of the work that's done inside the lung. In order to get the lung on this perfusion system to do ex vivo lung perfusion, we have to cannulate what's called the left atrium. So this is where the veins from the lungs return into the heart. And you can see there's no heart here. So we put this cannula on that tissue. We sew around the perimeter of it to make sure there's a tight seal. And then we cannulate the pulmonary artery. So you can see this is the main pulmonary artery here. And again, with no heart in the way, this is the vasculature that's connected. This goes into the left PA, into the right PA. And then you can see that other cannula that we did in the left atrium is already sewn in place and has sutures around the perimeter. And then we do what's called an intubation, where we'll tie this endotracheal tube or this breathing tube inside the trachea so that we can actually connect it to that ventilator that will keep the lungs moving and ventilating while it's on the system. So this is what one of our operating rooms looks like. This is a set of lungs that's in that plastic dome. You can see this is James. He's one of our specialists that works within our facility. You can see this is the reservoir. And then what you don't see below is the pump that actually perfuses this lung with that bloodless solution. The solution looks a little bit pink. Just because you can't 100% flush everything out of the lung, so there's still some residual blood that's left over in the lung, which turns the color of the perfusate pink. But this is what the lung looks like on the system. This is the ventilator that's connected with the endotracheal tube here, and this is where the lung sits for the period of time that we have it on the system. And there's a lid that comes over this part right here when we're not working on it. This is just a close-up of what the lungs look like when on the system. This is that pulmonary artery catheter that we had talked about earlier in the slides. This is the left atrial catheter, and then this is that breathing tube that's tied into place. This is the left lung. This is the right lung. And then you can see the vasculature here inside the hilum. I wanted to share some video, but unfortunately, anytime you share a screen with a presentation like this, the video can get a little bit choppy, so I didn't include the video, but this is a picture of what it looks like. And this is just another kind of dramatic photo of what it looks like on the system. And then during the time that we're working on the lungs, there's a camera inside this room that will do 360-degree views of the lungs. So the surgeons are logging in, they're reviewing the information, they're looking at the data, they're looking at the video of the lung. And then we also take what's called the bronchoscopy, which is this piece of equipment right here. And we actually go inside of this endotracheal tube and evaluate the inside of the lung, and the surgeons could see the video to all that as well. So this is all just information that helps them make a decision on whether the lung should go for transplant. The other thing that we do is do x-rays. So this is an x-ray that was done at hour 1 on the EVLP system. This is an x-ray that was done at hour 3. And you can see there's been some improvement from hour 1 to hour 3. So what does improvement look like when I look at an x-ray? Anytime you see areas on an x-ray that are white, that usually means it's not good. There's something that's keeping the penetration from going through the lung. So you like to see areas that are darker. So you can see this white area here at hour 1 is pretty obvious. But then when you look at hour 3, that area has been re-expanded and has improved in function. So this is a lung that actually improved while it was on our system. And once we finished the procedure, the lung was flown to the transplant center and was transplanted into a recipient that was on the transplant list waiting for a transplant. So we're a 24/7 operation. So we run 24 hours a day, 7 days a week. It doesn't matter when a transplant center calls us, we're ready to go. And our clinical staff, when we're on call, we're never more than an hour away from the facility, and we're ready to respond to any needs that exist within the transplant system in the US. And our goal is really to increase the amount of lungs available for transplants and to minimize the amount of patients that die every year waiting for a lung transplant. And again, just another picture of our facility in Silver Spring. And this is the team working on a lung right here. You can see the lung is kind of deflated, but this is what the operating room looks like. And in our facility in Maryland, there's 6 operating rooms that look like this. And then we have a command center in there, which is kind of nice. So it provides a live feed into each one of the procedure rooms. This command center gets pretty busy when we have lung procedures in-house. This is where people can watch the procedure. And this is kind of what the room looks like when there's a procedure that's live. You can see there's x-rays up, there's videos, and this is our IT specialist that processes all that information that's collected and sends it to the transplant surgeon, who will make a decision on whether the lung should go for transplant. And that's all I have for you.
Right. Very interesting, Sam. Can you tell us a little bit about your training and your background?
Yeah, sure. So my undergrad was in biology and chemistry. I ended up going to grad school at what was then the Medical College of Ohio in Toledo, and did a program in human donation science. And before coming to United Therapeutics Lung Bioengineering, I worked in the organ donation and transplant industry for about 8 years, facilitating the donation and transplant process for patients waiting in the state of Indiana.
All right. Very interesting. And I'm sure people learn quite a bit from this. And we're having a little trouble, unfortunately, getting it on Zoom, which we usually, on Facebook. So we are going to post it to Facebook later. We are recording, and we'll also possibly post it to YouTube tonight. But that was very interesting, Sam, and thank you so much for presenting.
Sure
thing. And Annie, is your team ready? All right, then I will be introducing Annie and her team.
Hi there. Can you hear me and see the screen?
Yes, we can.
That's great. Good afternoon, everybody. Thank you so much for having us today. Travis and Sam, that was fascinating, the kind of work you're doing. Thank you for sharing that with us. So we are from a biotech company called Viela Bio. We're really excited today to share with you our story behind discovering a drug that can really make a difference in the lives of many people living with chronic illness. So a little introduction about myself. My name is Annie Lau-Kilby. I'm a biologist. I'm particularly excited to learn about how our body's own immune system can fight infections and what happens when something goes wrong with that response. And this study of the immune cells makes me an immunologist. I received my PhD in immunology in Scotland, and then I've been spending the last 15 years here in the United States studying our immune systems. I'm really passionate about using this knowledge from the last 15 years every day at my job at Viela Bio as a scientist to develop new medicines for those in need. There's a few of us joining us from Viela Bio today who are pictured here. We have folks from many different sections of the company, so you can see how it really takes a team of both scientists and non-scientists to make new discoveries. So this slide is about Viela Bio. We are based at 3 different sites in Gaithersburg and Rockville in Maryland. Our focus, as I said, is to develop therapies for patients that are really in need, especially those that have developed autoimmune disease and inflammatory diseases. Recently, we received approval for an antibody called Uplizna, and that is what we specialize in. We develop antibodies as medicines. And this is really exciting for us when we got approval for Uplizna, and we'll be really happy today to share that journey with you. So first, we're going to go over the science. I'm going to hand over to my colleague, Nanette, who's a scientist in translational science.
Hi, everyone. My name is Nanette Mitterator, and I've been busy in the lab working towards making medicines to treat diseases like rheumatoid arthritis, lupus, and NMOSD, the disease that Annie just described, and other autoimmune diseases. Okay, let's jump into the science. So raise your hand if you've ever gotten a cold. And I'm going to assume, since I can't see any of you, that most of you have your hand up and have had a cold at some point. When you have a cold, it's caused by a virus. And the reason that you start to feel better is that antibodies that are made in your body fight this virus. So what are antibodies? Antibodies are Y-shaped proteins, as Travis described earlier. They're also called immunoglobulins that help stop intruders like viruses and bacteria from entering and harming your body. So how do these antibodies work? The first thing that happens is that a virus enters your body and you start to feel sick. Your immune system kicks in and causes your body's B cells to make antibodies. These antibodies then stick to proteins from viral antigens called antigens. Your T cells then attack these antigens that the antibodies have marked for it. So after the antigens have been destroyed, the cleanup crew gets busy to remove the infection by a process known as phagocytosis. After the infection is defeated and you're feeling better, antibodies stay in your body and are armed and ready the next time the virus wants to enter your body. So B cells and T cells, along with antibodies, are major players in your immune system to help fight infection. So Annie is going to take it from here.
Thank
You
you,
got it,
Nanette.
Annie?
Yeah, can you hear me?
Yes.
Great. Thank you, Nanette, for that introduction on antibody biology. So now you know about how our bodies use antibody to fight infection. We will discuss how the antibody is so important in our current COVID-19 pandemic. So in the last few months, you've probably heard of these three terms that are on this screen right now: coronavirus, SARS-CoV-2, and COVID-19. So here are some fun facts about these three terms. Coronavirus is actually a term used to describe a large family of many different RNA viruses. Scientists have actually been studying them since the 1960s. So we actually have built up 60 years of knowledge on this family of virus. And that is why scientists are able to develop vaccines and treatments so quickly right now. We're really working well together to do this. Interestingly, many of us have probably been infected with coronavirus before. It's because coronavirus is thought to cause 15% of all common cold. So that means every 100 people that have had a runny nose or a low fever or sore throat, around 15 of them is caused by coronavirus. We also know that coronavirus infects the respiratory tract. That means we breathe it in through our nose and it goes to our lungs where the infection occurs. So we breathe it in, and then when we get sick, we also breathe it out. And that is why it's really important to wear a mask. SARS-CoV-2 is a member of the coronavirus family. It's the virus that leads to COVID-19 disease. And it's actually very similar to SARS-CoV that caused a 2003 SARS outbreak. And that is why it's called SARS-CoV-2. They simply added a number 2 at the end because it's very similar to the virus back in 2003. So we actually know a lot about this virus. COVID-19 is the disease that it causes by the virus. So it develops symptoms like fever, chills, runny nose, cough, sore throat, headaches, very similar to the common cold, but some of it can get really serious, and that is why COVID-19 is a pandemic. So now you know about COVID-19, about antibodies. How do they play a role in COVID-19 infection? Well, as I said, the coronavirus, we breathe it in, it goes down our respiratory tract into our lungs, and that's where the virus infection occurs, in our lungs. When that happens, our immune system starts making antibody to destroy the virus. So as Nanette mentioned, we have these Y-shaped protein antibody that is made by our body that will bind to the virus and tell the immune system to, hey, the virus is here, come and destroy it. And this picture actually formed the basis of 2 tests that we currently use to test whether you have COVID-19. One of them is an antibody test. The antibody test measures the amount of antibody, so these Y-shaped proteins that are floating around in your blood. So it doesn't actually detect the virus, it detects how your body is responding to the virus. And it does take the body a few weeks to make these antibodies. So it's important to know that this antibody test does not actually tell you whether you have a virus infection right now. It just tells you you've had the infection and your body is responding to it. A second test you probably heard a lot about in the media is a PCR test. A PCR test works by detecting the virus by measuring the genetic material that's in the SARS-CoV virus. And they do this by doing swabs of your nose or asking you to spit in a tube and collecting your saliva. They do this because there's a lot of virus living in your nose and also in your mouth. So that way they can then look at the genetic material in these samples and then look at whether the virus is there. So this concept of antibody tagging the virus to destroy the virus is how our body works when everything is going well. However, sometimes our immune system goes awry and we make antibody against our own body tissues rather than any invading virus and infections. And this is what our company is very passionate about, developing medicines about autoimmune diseases when our own body starts attacking ourselves. Some examples here: type 1 diabetes, rheumatoid arthritis, lupus, multiple sclerosis, are all examples of autoimmune diseases where we start attacking our own body. And with that, I'm going to hand over to Shauna, our clinical scientist, about how we develop drugs for these autoimmune diseases.
Thanks, Annie. Hi, everyone. My name is Shauna Quinn, and I'm a clinical scientist with Viela Bio. I'm an immunologist by training, and my passion has always been understanding and treating autoimmune diseases. As Annie mentioned, this occurs when parts of the body that normally fight infection become dysregulated, and they attack parts of our body instead of invading pathogens, for instance, COVID. Viela focuses on developing drugs for people with autoimmune diseases. An example of this is NMOSD. As Annie mentioned, the FDA recently approved our drug Uplizna for treatment in these patients. This disease is where the body attacks the optic nerve and the spinal cord, causing paralysis. So what are the steps involved in getting a drug approved and available to patients? Next slide, Annie. Well, we first have to start at the beginning, of course, with basic research and early discovery into specific disease areas or drugs of interest. This usually takes place in vitro, which means outside of an organism or living thing. And for instance, this could be in a Petri dish or test tube. Next, we would move on to in vivo or preclinical testing. And this usually takes place in animals, for instance, mice, to answer basic safety questions. If we are happy with the safety, we move on to the clinical development stage, which is the area that I work in, which is human testing. The first in human or phase 1 is where a small group of volunteers receive the drug in order to test safety. Remember, before this step, the drug would only have been tested in animals. If phase 1 is successful, we move on to phase 2, which usually is in a larger population, possibly in a particular disease population, and again looks at safety, but also some efficacy of how the drug is working in these patients. And phase 3 is usually the final step before FDA approval and tests the drug in a much larger, more diverse population. During the clinical development process, you can see there are a huge number of people needed, including patients, scientists, doctors, and nurses. But of course, this is only to name but a few. If the phase 3 is successful, the FDA will review all safety and efficacy related data and determine if the drug will be approved. Next slide. So how many drugs get approved? Is it every single one that goes through this process? In actual fact, very few compounds and drugs get approved. If you look at this diagram, for every 10,000 compounds that go through the drug discovery, you can remember this is the part that is in vitro or before it goes into any living organism. This will actually only 250 of those compounds would go into preclinical development. Again, animal models testing this drug. And of those 250, only 5 would ever make it into testing in human, into the clinical development stage. And remember, this clinical development stage includes your phase 1, your phase 2, and phase 3 clinical studies. So do you have to be a scientist to get involved in the drug development process? Absolutely not. Next slide, Annie, please. You can see here that it takes a huge effort from all different types of teams in a company like Viela in order to get one drug approved. You can get involved even if you're a scientist or non-scientist, and you can be a member of, say, for instance, clinical development, clinical operations, medical affairs, research, even business development, commercial, regulatory affairs, office management. The list goes on. It really takes an army and more to get a drug approved. So now I'll hand you over to Jennifer. I think we also just have a little bit about what's next in our UPLIZNA. So UPLIZNA was just recently approved by the FDA. So this means that NMOSD patients can now be prescribed and treated only in the US. So now we will file for approval in the EU and Japan. And what's really important is as we enroll more patients to be treated with NMOSD, with UPLIZNA for NMOSD, we will continue to collect safety data, and this is really important. So thank you all for listening, and I'll now hand you over to Jennifer Cunningham, the Senior Office Manager, to discuss other exciting aspects of being a member of the Viela Bio family.
Thanks, Shauna, for the introduction. I'm Jennifer Cunningham, Viela Bio Senior Office Manager, as well as the Senior Executive Assistant to the CEO of Viela. My job is to manage the administrative staff. And my staff in support of Viela, we support the scientists, we support other departments, as well as we participate in the onboarding of all new hires to the company with helping the HR team. As part of that duty, or as part of my duties, I help set up new hires, including the badging and the training links associated with each new hire. We have 3 offices, 2 of which are labs, as noted earlier. One lab is the research and development, where the scientists who are on this in this festival work. And then we have another lab that's located in Rockville, and that is our commercial lab. I have 3 members under my team who also help me manage the 3 offices. Viela Bio is a team who also likes to celebrate special occasions and milestones, and most noteworthy milestones like our 6-month, our 1-year, and our 2-year anniversaries of the inception of our company, as well as throwing fun occasions like holiday parties. And we like to welcome the newest, littlest members to our team with baby showers. Last but never least, we like to give back to our community through volunteer service. Most recently, Viela Bio volunteered at Comfort Cases preparing backpacks for those children in foster care. We also did an opportunity where we made snacks for the parents of children who are receiving specialized treatment at the Children's Inn in Bethesda. We also participated in the STEM Festival last year. We believe it is important to teach the next generation of scientists. Viela Bio also believes in doing our part to always give back to the community.
So thank you everybody for joining us today. I hope we provided you with an overview of a biotech company and highlighted the many career options possible for both scientists and non-scientists. And the most important thing I think to take home is that as a team, we are really passionate about discovering medicines for those in need. So with that, feel free to reach out to me anytime through Phil. Thank you.
All right, thank you, Annie, and your team for that great presentation. So we'll be wrapping up in just a moment. First, excuse me, I have put in the chat box the link to our survey. We'd appreciate it if you complete the survey. Let us know what you thought about this program. Of course, check out our website to see the rest of the events. Last Friday, which you can find on our Facebook channel, we had a program on biotechnology on prosthetics from a company, Dankmeyer, that has been making prosthetics for 65 years. So if you're interested in that, please check it out on our Facebook page last Friday, and it'll be on our YouTube channel after the festival. And if you have any questions for any of the teams, please put them in the chat quickly or email them to me at mdstemfest@gmail.com. And again, thank everybody for both participating and for joining the program. And I hope you learned something, and I hope you'll strongly consider a biotech field. So thank you. Thank you very much, everybody. Have a great day, and we hope to see you soon. Bye-bye.
Thanks, everyone. Bye.
Thank you, everybody. Bye-bye.
Thank you.